This is the third lecture in Unit 6 for microbiology. This section is going to focus on the eukaryotes. There are a few options for reproduction in the eukaryotes. These include asexual reproduction, binary fission, budding, fragmentation, spore formation, schizogamy, and sexual reproduction. Since the eukaryotes have a nucleus, we must divide the nucleus. When there is a single copy of the chromosome, the cell is haploid. When there are two sets of the chromosomes, the cell is diploid. Mitosis is the process of asexual cell division. It begins with prophase, where the chromosome is formed. The nuclear envelope disintegrates and the mitotic spindle begins to form. In metaphase, the chromosomes align along the mitotic spindle in the center. During anaphase, the chromatids separate. We also see cytokinesis and the formation of a cleavage furrow. In telophase, we have two new daughter cells where the chromosomes unwind to form chromatin and a new nuclear envelope forms. There's a video link here that shows the process of mitosis if you would like to watch that on your own. In meiosis, we have nuclear division into 4 nuclei, each haploid. This is sexual reproduction. In early prophase 1, the homologous chromosomes pair, and in late prophase, we have crossing over for genetic variation. In metaphase 1, the chromosome tetrads align along the mitotic spindle. During anaphase 1, the chromosome tetrads separate. In telophase 1, we have 2 diploid cells. And here we've got this diagram embedded in the notes that shows this process happening. The nuclear envelope disappears and a new mitotic spindle forms in prophase 2. In metaphase 2, the chromosomes align and the sister chromatids separate in anaphase 2. In telophase 2, we have 4 daughter nuclei in 4 haploid cells. In the second division, there is not a second copy of the chromosomes, leaving haploid cells. In cytokinesis, we have the division of the cytoplasm. Typically, this begins in anaphase and is completed in telophase. If it does not occur, it will produce multinucleate cells known as koinocytes. And there's also a video here showing meiosis. With schizogony, we have multiple mitosis to form a multinucleate schizont. The cytokinesis occurs producing numerous uninucleate daughter merozoites. This occurs in the plasmodium. So you can see in this picture the schizogony in there. We also have a couple of videos of malaria schizogony occurring. Classification started by grouping organisms based on observable traits. Now classification is moving towards schemes based on genetic relatedness. Because this technology is fairly recent, you will see classifications of some organisms changing. You will also notice that's going to be what's most likely to change in a new textbook. Although the classification is changing in the textbooks, out in the field, in the workplace, the classification changes don't always catch up. So in this class, I'm not too worried about you knowing the latest current classification, and for the most part, the classification doesn't have anything to do with how they behave in a clinical presentation. The protozoa are eukaryotic, unicellular organisms lacking a cell wall. Most require moist environments because without a cell wall they would desiccate. They often live in moist soil or sand, decaying organic matter, and a few are pathogens. Their morphology is varied. Some of the ciliates have 2 nuclei and some lack mitochondria. The trophozoite is the motile, free-living feeding stage. And then the cyst stage has a capsule and low metabolism, making it more resistant. Most are chemoheterotrophic, and most reproduce asexually, but but by binary fission, schizogony, and then some sexual production is seen. Gametes are formed in those who reproduce sexually. The alveolates have a small membrane-bound cavity called alveoli. The ciliates have cilia, which may cover the surface or just occur as a few tufts. All ciliates have 2 nuclei. The Vorticella uses a whirlpool current to move towards its mouth. The Valentidium is pathogenic, and the Didinium is able to phagocytize other protozoa. There's a video here that you can watch of the Vorticella spinning the cilia to actually move food towards their mouth if you'd like. The apicomplexans have special intracellular organelles. All of these are pathogens and include the Plasmodium species that cause malaria, Babesia causing anemia, and Toxoplasma causing toxoplasmosis. The Dinoflagellates are unicellular organisms with photosynthetic pigments. They have plates of silica, lack histones, and are found in freshwater or marine plankton. A lot of information on these is actually, you can see more with oceanography websites. Many are bioluminescent and others are red. These are the organisms responsible for the red tide because they can make a neurotoxin that poisons humans. The red tide is a concern when eating shellfish obtained during tides high in dinoflagellates. Amoebas are protozoa, which move and feed by pseudopodia. They lack mitochondria and divide by binary fission. The radiolarians and foraminifera are amoeba with sheets of silica and calcium carbonate. The pathogenic amoeba are the Naegleria, acanthamoeba, and entamoeba. So we've got information on these pathogenic amoeba. These are the ones that occasionally come up that people have acquired while swimming in lakes. And usually they're not problematic unless they get into the brain. And then most of the time, if you get an amoeba infection in your brain, you're going to die. The Euglenozoa have characteristics of both plants and animals. The euglenoids are unicellular photoautotrophs with chlorophyll A and B and carotene. They store food as paramylon and lack cell walls. They have flagella and can form cysts. The kinetoplastids have a single large mitochondrion. The kinetoplast is the region of the mitochondrial DNA. Some are pathogens, such as the trypanosomas, causing African sleeping sickness and Chagas disease. So there's a video here if you'd like to see more on those. The videos tend to be a little more interesting than the websites were. The Archaeosa lack mitochondria, Golgi bodies, and peroxisomes. These include Diplomonadia and the Parabasilla. The Diplomonadia have 2 nuclei and multiple flagella. Giardia is an organism that forms resistant cysts. And there is some information here regarding Giardia. Giardia is found all over the world in water supplies. If you're out in the woods, it's always best to assume that the water supply has Giardia in it. In any case, it's just that abundant. The Microsporidia have polar filaments and are obligatory intracellular parasites. The noseoma and microsporidium can cause disease in the immunocompromised. The Parabasilla lack mitochondria, have a single nucleus, and a parabasal body. The Trichonympha live in termite guts and allow them to digest wood. The Trichomonas causes vaginitis in females but is asymptomatic in males. Fungi include mold, mushrooms, and yeasts. Their cell walls have chitin, and they are chemoheterotrophs, which decompose dead organisms. Their morphology is more complex than prokaryotes. The thallus is the vegetative or non-reproductive body of the fungi, with a great deal of variation. The hyphae are long-branched, tubular filaments in mold. They are said to be septate when the hyphae are divided into cells by cross walls. They are aseptate if the hyphae do not have cross walls and instead are multinucleate or quinocytic. Some are dimorphic and produce two types of thalli. The mycelium is a tangled mass of intertwined hyphae on mold. This is what appears as the fuzzy part of mold. So with them being dimorphic, it can make identification of them require a little bit more memorization of them. Fungi are saprophytes that absorb nutrients from the remnants of dead organisms. The hausteria are modified hyphae that penetrate into the tissues to withdraw nutrients from living plants and animals. When you cut the visible mold off of food, you often miss the hausteria that extend in further than what is visible to the naked eye. So when you slice that mold off the cheese, unless you're cutting back quite a ways, you're still eating some of the mold. Budding is the asexual reproduction used by yeasts. They bud similar to prokaryotic cells. In some species like Candida albicans, series of buds remain attached to make pseudohyphae. Some molds will make asexual spores. The sporangiospores form a sac sporangium. The chlamydospores form thickened walls inside the hyphae. The conidiospores are produced at the tips or the sides of the hyphae, but not within the sac. There are many types of conidia, including the arthroconidia, blastoconidia, and conidiophores. Some fungi will have sexual spore formation. They form haploid cells from a positive and negative thalli, instead of calling them male and female, which fuse to form a dikaryon. It is described as an N plus N cell. The two nuclei fuse to form a single 2N nucleus. Meiosis of the second nucleus restores the haploid state, and they are partitioned into positive and negative spores. There are three classes of fungi. The division Zygomycota are the coenocytic molds with 1,100 species. The zygomycetes do asexual reproduction via sporangiospores to form sexual zygosporangia. They usually develop from sexually compatible hyphae tips, as in the rhizopus. The division Ascomycota contain 32,000 species of molds and yeasts. The Asco spores are haploid and form in sacs called asci. The asci are in fruiting bodies called ascocarps and reproduce asexually by conidiospores. They will spoil food and act as a plant pathogen. The Caviceps purpurea grow on grain and produce LSD, the lysergic acid diethylamide. Penicillium mold and truffles are additional examples. The Saccharomyces ferments sugar to ethanol. The Basidiomycota have 22,000 known species. The basidiocarps have fruiting bodies and are tightly woven hyphae that extend into club-shaped basidia, which produce sexual basidiospores. Many are decomposers and hallucinatory. They include rusts and smuts that affect crops. A couple of examples are the amanita or deathcap mushroom and Cryptococcus neoformans causing meningitis. The Deuteromycetes are being reclassified using rRNA, and it was determined most in this category actually belong in the Ascomycota. Sexual stages are unknown, and several are pathogens. A common example is the trichophyton, which is the cause of ringworm. To get more into the details of what goes on with the fungi and the diseases, those are usually covered in mycology class and is enough information to be a whole separate class in itself. The lichens form a partnership between fungi and photosynthetic microbes. The hyphae of the fungi surround the photosynthetic cells and provide nutrients, water, and protection from desiccation and harsh light. The algae or cyanobacteria provide the fungus carbohydrate and oxygen from photosynthesis. In some lichens, the phototroph releases 60% of the carbohydrate to the lichen. Not all of these relationships are mutually beneficial. There are 14,000 species of lichens in 3 shapes. The crustose shape grows attached to a substrate and may extend into the substrate several millimeters. The foliose shape is leaf-like, and the margins grow free of a substrate. The fruticose are erect or hanging cylinders. If you run into lichens, you'll find that they're really hard to remove off of trees a lot of times because the substrate, they do extend down into the substrate of the tree. There's a couple of websites here. One of them shows the lichens growing in a video. The algae are simple eukaryotic phototrophs which use chlorophyll A. Phycology is the study of algae. Most live in aquatic photic zones where light is available. Others who live in diverse habitats have accessory photosynthetic pigments to trap short wavelength light in deeper water. Phycoerythrin is a red pigment found in the red algae. They are unicellular or colonial and may have simple or multicellular bodies called thalli. They grow in branched filaments or sheets, and some will have pneumocysts, which are gas bubbles allowing them to float. Unicellular organisms can do asexual reproduction with mitosis and cytokinesis, or sexual reproduction with gametes fusing to form a zygoid for meiosis. The multicellular organisms may do asexual reproduction by fragmentation and spore formation. In sexual reproduction, every cell becomes a gamete. Alternation of generations occurs with alternating between haploid and diploid cells. Studying the reproductive cycles of these organisms can be a pretty diverse topic to cover, and goes into a lot more detail than what we're presenting here. Remember, this is just an overview of the different organisms that are out there. Classification of algae is not yet settled, but so far is based on the color of the photosynthetic pigment. The chlorophyta or green algae have many similarities to plants. They have both chlorophyll A and B, and sugar and starch reserves. Many have cellulose walls and were placed in the kingdom Plantae in the past. They may be unicellular or multicellular, freshwater or marine, and include the Protheca, Codium, and Trabuxia. The Rota Fighter or red algae have the red pigment Phycoerythrin. They store glycogen as fluoridine, have starch in the walls, and most are used in making agar or carrageenan. They range in color. The male gametes form non-motile spermatia. Most are marine, but some live in fresh waters. Examples include chondrus and gelidium. The phaeophyta are the brown algae. They are in the kingdom Straminophila. They have chlorophyll A and C, carotene, and a brown pigment xanthophytus. Most are marine. The gametes and spores are motile. These algae use the polysaccharide laminarin and oils as food reserves. The walls have cellulose and alginate, which are used as thickener in foods. So the agar we use in class is going to involve the use of algae. And then a lot of the thickeners you have in your foods, such as things like ice cream, are actually going to come from algae. The chrysophyta include the golden algae, yellow-green algae, and diatoms. All have carotene in greater abundance than chlorophyll and use the polysaccharide crystalmarin as a storage product. They have diverse cell walls and pigments, and some will lack cell walls. Others have ornate coverings. The diatoms have two frustules that fit together like petri dishes and are a component of plankton. They are usually used in polishing compounds, gardens, and detergents and paint. So you can buy diatomaceous earth that has greater amounts of diatoms in it. The water molds are not true fungi. They have tubular cristae in their mitochondria, cellulose walls, and two flagella. They have a true diploid thalli. They can decompose dead animals and may damage crops. Phytophthora kills potato crops. The slime molds lack cell walls and are phagocytic. The plasmodial slime mold or acellular slime molds include the Fusarium. They exist as streaming, coenocytic, colorful filaments of cytoplasm that live in forest litter. The cellular slime molds exist as individual haploid mixamoeba. They phagocytize bacteria, yeast, dung, and decaying vegetation. So there's a time-lapse video here of a slime mold moving if you're not sure what slime molds look like. There are a few other eukaryotes related to microbiology that are not always microscopic. The helminths are parasitic worms, which are microscopic in the infective and diagnostic stages. The arthropod vectors are animals with segmented bodies, hard exoskeletons, and jointed legs that carry pathogens. Arachnids have 8 legs and include ticks and mites as vectors. Spiders do not transmit diseases. However, if a spider bite itches, you can always give yourself a secondary infection. The insects have 3 pairs of legs, a head, thorax, and abdomen. They are the greatest number of vectors. With flight, they have a broader home range and are hard to control. These include fleas, lice, flies, true bugs, and mosquitoes. And here's a few websites on here on the helminths, the parasitic worms in there. If you are interested in more information on those, there's a video on properly removing a tick, and then there's a lot of information out there on the insects. Most of the information that you find on insects is usually going to be on ways to control them to try and reduce the amount of insect causing disease.